In Chapter 8, we're going to be looking at metabolism. We're going to also be looking at the laws of thermodynamics, free energy change, the types of work a cell does, how enzymes speed up reactions, and how enzyme activity is controlled. Cellular respiration extracts energy that's stored in sugars and other fuels. Cells apply this energy to perform work. Here we see a picture of a termite mound. On that termite mound are beetle larvae that are glowing. These beetle larvae are converting stored energy to light, which is why they are glowing. Metabolism refers to all of an organism's chemical reactions, everything that's going on. A metabolic pathway begins with a specific molecule and ends with a product. And each step in getting to that final product is catalyzed or helped along by a specific enzyme. So here we are starting with molecule A and we want to get to product D. So we have A, enzyme 1 helps to get to B, enzyme 2 to C, and an enzyme 3 to the final product of D. There are two kinds of metabolic pathways, catabolic and anabolic. Catabolic pathways release energy by breaking down complex molecules into simpler ones. So for example, cellular respiration, it's the breakdown of glucose in the presence of oxygen to carbon dioxide and water. Anabolic pathways involve the consumption of energy, so they use energy to build complex molecules from simpler ones. An example of this is making a protein from amino acids. Studying how energy flows through living systems is what we call bioenergetics. And it's important to know that the energy released from catabolic pathways drives the anabolic pathways. We've used the term energy a bit here. Energy is the capacity to cause change. Usually we look at it in regards to the ability to do work. One type of energy is kinetic energy. This is the energy associated with the motion of objects. So for example, using a cue stick to push a ball on a pool table. Thermal energy is a type of kinetic energy, and it's the type that's associated with random movement of atoms or molecules. It's energy in its most random form. Potential energy is the energy matter possesses because of location or structure, what we typically think of as stored energy. Chemical energy is energy available for release in a chemical reaction. It's a form of potential energy. So for example, glucose, which can be broken down, has high chemical energy. Energy is able to be converted from one form to another. In figure 8.2, we see transformations between potential and kinetic energy. So on the top left, we see a diver on a diving board. He has more potential energy on the platform than in the water, and that's due to his location. It's up high. To the right, when he dives, that potential energy is converted to kinetic energy, the energy of movement. Once the diver hits the water and surfaces, he has less potential energy in the water than on the platform. And when he climbs up the stairs to the diving board, he's converting the kinetic energy of muscle movement to potential energy. Thermodynamics is a study of energy transformations in the collection of matter. One type of system, and we talk about system, we mean a collection of matter we're interested in. One type is an isolated system. This is a system that is unable to exchange energy or matter with its surroundings. So an example of this would be a liquid in a thermos. A liquid cannot exchange energy with the air outside of the thermos. The other type of system is an open system. In this type of system, energy and matter can be transferred between the system and its surroundings. So living organisms are what are considered open systems. Now there are different laws that govern thermodynamics. We're going to look at two of them. According to the first law of thermodynamics, energy of the universe is constant. It can be transferred and transformed, but it cannot be created or destroyed. It's also called the principle of conservation of energy. So, for example, the electric company doesn't make energy, it converts it to a convenient form. So, on the left hand side, we see an example of a bear eating fish. He's converting that chemical energy in food to kinetic energy and other forms. During every energy transfer or transformation from one form to another, some energy is going to be unusable. This is often lost as heat. So according to the second law of thermodynamics, every energy transfer or transformation increases the entropy of the universe. And entropy is a measure of disorder or randomness. So in this example, down below, we have the bear. He's using that chemical energy of the food to convert it into kinetic energy. And some of that energy is going to be released to the environment as heat. That's increasing the molecular disorder of the environment. Molecules are being released in the form of heat. Living cells convert organized forms of energy to heat. And again, like we said, heat is a more disordered form of energy. A process can be classified as either spontaneous or non-spontaneous. A spontaneous process occurs without any energy having to be put in. This type of process leads to an increase in entropy. It's energetically favorable. Processes that decrease entropy are non-spontaneous. They will only occur if energy is provided. So spontaneous will occur without any energy being put in. Spontaneous need energy to be put in order to occur. Biologists want to be able to figure out which reactions occur spontaneously and which do not, which ones require energy to be put in. So we look at a term that's called free energy. This is the portion of a system's energy that can do work. So what we do is we compare the free energy at the beginning of the reaction to the free energy at the end of the reaction. And we use the term delta G, which is the change in the free energy. We use G to represent free energy, and that lets us know the difference. And to get that, we just take the free energy at the final state and subtract the free energy initially. If a process is spontaneous, the change in free energy is negative. Energy is not needed to be put in. During a spontaneous change, the free energy decreases and the stability of a system increases. It has less free energy available, so it's becoming more stable. Figure 8.5 shows the relationship of free energy to stability, work capacity, and spontaneous change. So let's look at the first process, which is looking at gravitational motion. So we have the diver on top of the diving board. When he's on top of the diving board, he has a high free energy. The system is less stable, and there's a greater work capacity. Now again, a spontaneous change does not need energy to be put in. So the free energy of the system decreases. The system becomes more stable. The released free energy can be harnessed to do work. At the very end, once the diver has made his dive, there's less free energy. The system is more stable, and there's less work capacity because there's less free energy available. Chemical reactions can be classified based on the change in free energy. So one type of reaction is an exergonic reaction. This involves the release of free energy. This is spontaneous, and the delta G is less than 0. Remember, the change in free energy we get by subtracting the initial energy from the final energy. So if you look in panel A for an exergonic reaction, on the x-axis we have progress of the reaction, and at the y-axis we have free energy. So if you look, the reactants have a lot of free energy. No energy has to be put in. Some energy is going to be released. The products at the end have less free energy. So if we take the energy of the products and subtract the energy of the reactants, that delta G is going to be less than 0. In an endergonic reaction, free energy is absorbed from the surroundings, so energy has to be put in. This is non-spontaneous, and its change in G or delta G is greater than 0. So this graph is set up the same way, but if you look, energy is being put into the reaction. The initial reactants have less free energy than the products do at the end. So when you take the energy of the products and subtract the energy of the reactants, it's still a positive value. So the change in G is greater than 0. This is an important slide to understand the difference between exergonic and endergonic, which requires energy, which is spontaneous versus non-spontaneous, and if the delta G is positive or negative. There are three main types of work that a cell can do. One is chemical work. This is pushing an endergonic reaction like we just looked at that would not occur spontaneously. So, for example, making a polymer, making a protein from amino acids. A second type of work is called transport work. So pumping substances across membranes against the direction of spontaneous movement. So we looked at that previously when we were talking about active transport. And a final type is mechanical work. And an example of this would be the contraction of muscle cells. Now, to do work, cells manage energy resources by energy coupling. That means they use an exergonic process to drive an endergonic process. And this is usually controlled by ATP. ATP is something I mentioned in a previous chapter. So again, ATP is adenosine triphosphate, and this is the cell's energy shuttle. And in figure 8.9a, we see the structure. So on the right-hand side, you'll see adenine. That's a base. Attached to it is ribose, a sugar, and attached to the sugar are 3 phosphate groups. So that's why it's called triphosphate. Adenosine triphosphate. The bonds between the phosphate groups of ATP's tail can be broken by hydrolysis. Remember, hydrolysis is when water is added to break a bond. This is exergonic. Energy is released from ATP when that terminal or end phosphate bond is broken. So in this diagram in 8.9b, we see the hydrolysis of ATP. We have the molecule ATP. Water is added and one of those phosphate groups is released. At the same time, energy is released. That energy that can then be used to perform work. Energy from the exergonic reaction of ATP hydrolysis can be used to drive an endergonic reaction. Overall, the coupled reactions are exergonic. ATP drives endergonic reactions by phosphorylation, which just means transferring a phosphate group to another molecule. We saw that in the previous chapter with the sodium-potassium pump. The recipient molecule is now called a phosphorylated intermediate. For the time being, intermediate, it has a phosphate group attached to it. It's phosphorylated. Now this reaction, this looks complicated. I just want you to look at the delta G values. So in the top panel for A, we have a delta G value of plus 3.4. In the bottom panel, C, there's a delta G value of negative 7.3. When we add these up, we get a total delta G of negative 3.9. So this is just to show when you couple the reactions together, the overall delta G is going to be negative. It's going to be an exergonic reaction in total when these reactions are coupled together. Transport and mechanical work in the cell are also powered by ATP hydrolysis, breaking off that final phosphate group from ATP. ATP hydrolysis leads to a change in protein shape and binding ability. And this example below looks like what we talked about with the sodium-potassium pump. Remember, a phosphate group broke off and the protein changed shape in order to move one solute from one side of the cell to the other. An organism at work uses ATP continuously. ATP is a renewable resource that is regenerated by the addition of a phosphate group to ADP, adenosine diphosphate. The energy to phosphorylate ADP comes from catabolic reactions in the cell. So in this example down below, we see the energy coming in from catabolism. Remember, catabolism is exergonic, it's energy releasing, and this energy is used to add the phosphate group to ADP to produce ATP. Water then breaks down ATP and energy is released. And an example of the importance of ATP regeneration we can see in the muscle cell. The muscle cell consumes and regenerates 10 million ATP molecules per second. Now, if you remember from previous chapters, an enzyme is a catalytic protein. That means that it can speed up a reaction without itself being used up. The initial energy needed to start a chemical reaction is what we call activation energy, or sometimes energy of activation, represented by the capital letter E and a subscript of A. We're going to see that enzymes work by lowering this activation energy. Here we see the changes in energy in an exergonic reaction. Now, oftentimes something doesn't go just from reactants to products. There's a transition step where there's some intermediates formed. And we can tell this is exergonic because the free energy of the products is less than the free energy of the reactants. However, even though it's exergonic, you'll see that a little bit of energy is needed to get it to that transition portion, that transition state. So when an enzyme speeds up a reaction, how does it do this? It does this by lowering that energy of activation, that little bump that's needed to get the reaction going. So here we have the progress of the reaction on the x-axis and the free energy on the y-axis. The black is showing how the reaction would go without an enzyme being added. The black arrow on the right shows the amount of energy of activation needed without the enzyme. The red line shows how the reaction would proceed with an enzyme being added. And you can see from that little red arrow, the energy of activation that's needed when the enzyme is present is lower. So the enzyme works by lowering the energy of activation. It doesn't need as much energy in that little initial bump to get the whole reaction going. The reactant and enzyme axon is called the enzyme's substrate. The active site is the region on the enzyme where the substrate binds. It's very specific. So remember, an active site is something on the enzyme. Induced fit is when there's a change in the shape of the active site when the substrate first binds so it can bind more tightly. So the substrate binds in the active site, it recognizes it, and then it grabs onto it a little more tightly. Figure 8.16 shows the active site and catalytic cycle of an enzyme. So in step 1, we see the substrates entering the active site. In step 2, we see the substrates are held in the active site, and these are just weak interactions. When that happens, the active site lowers the energy of activation. Then the substrates are converted to products. The products are released, and now that active site is available again for new substrates. An enzyme's activity can be affected by environmental conditions. So, for example, things such as temperature, pH, and chemicals. Each enzyme has an optimal value for each of those that it works best at. So in panel A, we see the optimal temperature for 2 enzymes. So on the left in black is the optimal temperature for a typical human enzyme. It usually works best at 37 degrees Celsius. On the right in red, we see the optimal temperature for an enzyme of a thermophilic, which is a heat-loving bacteria, which is more than twice the temperature, 75 degrees Celsius. In the bottom left, we see an example of a geyser. This is one place where you would find these thermophilic or heat-loving bacteria. In the bottom panel, we see how pH optimums can vary. So, for example, on the left, we see the optimum pH for pepsin, a stomach enzyme, is around 2. The optimal pH for trypsin, an intestinal enzyme, is around 8. So again, these optimum values are going to vary depending upon the enzyme. Sometimes enzymes need a little bit of help. A cofactor is a non-protein enzyme helper. They can be inorganic or they can be organic. If it's organic, this is what we call a coenzyme. Coenzymes include vitamins. There are some things that can inhibit enzymes. So some inhibitors covalently bond to enzymes and can be irreversible. However, there are also reversible inhibitors. We're going to look at two categories. One category is a competitive inhibitor. This binds to an active site of an enzyme. It's competing with the substrate for access to the active site, which is called a competitive inhibitor, and this reduces the activity of the enzyme. The other possibility is what's called a non-competitive inhibitor. It does not bind to the active site, it binds to another part of the enzyme, but this causes the enzyme to change shape and making the active site less effective. Some examples of inhibitors include toxins, poisons, pesticides, and antibiotics. One example is penicillin. So what penicillin does, it blocks the active site of an enzyme that many bacteria use to make cell walls. Figure 8.18 shows the inhibition of enzyme activity. So in A, we see normal binding. We have an enzyme, we have a normal active site, the substrate binds to it. In B, we have competitive inhibition, where we have a competitive inhibitor, it binds to the active site, so now the substrate cannot bind in there, can't get in there. And in C, we have a non-competitive inhibitor. So the non-competitive inhibitor binds somewhere other than the active site, but it changes its shape and makes it less effective. Regulation of enzyme activity helps control metabolism. If a cell's metabolic pathways were not tightly controlled, there would be chaos in the body. And one form, like we just looked at, was allosteric regulation. And this is when a regulatory molecule binds to a protein at one site and affects the protein's function at another site. And again, these can be inhibitors. They could also be activators to help a process occur. The last type of enzyme control we're going to look at is what's called feedback inhibition. This is when the end product of a metabolic pathway shuts down that pathway. So if you look at the purple enzyme where it says active site available, that's our enzyme. Normally, a substrate will bind to it. It goes through different steps, and at the bottom, you see in pink, we have our end product. Now, once we get a high enough concentration of that end product, it's going to bind to that initial enzyme that was used to help create it. It's going to change its shape so the active site isn't available, so that substrate cannot bind in there again. So the end product is controlling whether more of it is made or not.